Hi everyone, my name is Esther Yoon. I'm an MD PhD student in the Carville Lab at Northwestern University, and today I'd like to present to you about my work on reading between the lines of RNA. And first, I'd like to tell you the story of Emma. Pictured here on the left is Emma, and to the right is her very supportive family, her dad Brian, her mom Julia, and her sister Claire. I first met Emma at the start of my PhD. When I met Emma, she was 5 years old, she couldn't talk, she couldn't walk, and she was in a wheelchair. And her family had spent years searching for a diagnosis, going from doctor to doctor across the country, but no one could give them an answer. So they came to our lab, the Carville Lab, as experts in identifying the genetic causes of diseases. So what we did is we sequenced all 3.1 billion DNA bases of Emma's DNA, and what we found is that she has a piece of DNA missing, specifically in this area called chaser. So all of us right now sitting in this room, we have this gene called chaser, but in Emma's, this chunk was missing. And as a quick refresher from high school biology, the central dogma, I'd like to show you this piece of DNA, or just DNA in generally, which are this essential molecules that contain really important information to make up our bodies. And DNA gets made into RNA, which are these intermediary molecules that eventually go on to make protein, what we like to think of as these workhorse molecules in the cell that do the actual work and make up our skin and hair. And what's interesting about chaser is that while the chaser DNA goes on to make RNA, it actually doesn't make any protein. So a lot of people actually thought the missing piece in Emma and chaser actually didn't do anything because there's no protein to be made. But I wanted to think differently. And what I think is that the non-coding RNA, which give structure to our story. So if you think about the entire human DNA like a book, then you can consider the coding parts of our RNA like the letters O and F that make words like of and spaces, like protein, that give meaning to the book. And the non-coding RNA, you can consider like the spaces, the punctuations, or the lines that delineate the book. And while the non-coding RNA doesn't make any words, you can see that when the non-coding RNA is gone, we lose sense of the story. So I wanted to see if chaser was actually important for what was causing Emma's disease. So to study this, I took cells directly from Emma's skin and blood and studied them under the microscope. And what I had found is that chaser is essential in controlling the amount of CHD2 produced, which is a very important brain protein. So as you can see here, you can consider this as a healthy cell. Right next to chaser is another DNA called CHD2. And it does follow the central dogma, as in it goes from DNA to RNA to this protein. And what we found in the lab is that the CHD2 protein is really important in both developing and maintaining a healthy brain. And what I found is that chaser DNA, while it doesn't make any protein, it does make RNA. And in the RNA state, it recruits a team of proteins at the right place and at the right time to control CHD2 levels. And so you can see here are a bunch of different proteins that are all over the cell. And what chaser RNA does is it recruits them right next to the CHD2 DNA. And that when it monitors and sees that there's a lot of the CHD2 protein produced, then it stops CHD2 from getting made. And what I have found in Emma cells is that because she lacks this chaser to recruit these proteins, CHD2 protein levels cannot be monitored, and so that leads to too much CHD2 in her cells. And I found that having too much of this protein is bad and what caused her brain dysfunction and ultimately her disease. And similarly, we had identified 2 other individuals who have a deletion in chaser and found them also to have a very severe disease with too much CHD2 in their cells. And with that, we were able to recognize chaser deletion as an official disease. And I'd just like to highlight that Brian, Emma's dad, is featured as an author in the study because without his dedication to find an answer for his daughter, all these clinicians and scientists wouldn't have come together to make this discovery. And so with this finding, I am trying to find a drug target now to figure out if we can cure patients with chaser deletion. And so we know that the root cause of disease in these patients is having too much CHD2. So what I'm doing is I'm using something called antisense oligonucleotides. They're a mouthful, so we just call them ASOs, which are these synthetic molecules that can target a specific RNA and degrade it. So I've designed an ASO that can target the CHD2 RNA, and which ultimately degrades the CHD2 protein and restoring CHD2 levels down in chaser deletion patient cells. And while I've found this to be effective in MS cells now, it will probably realistically take years to get into clinical trial. And what I like to say is that we have learned the central dogma, but it really only applies to 1% of our RNA, and the rest, the 99%, is actually non-coding. And chaser is only one of this many of the non-coding RNA. So what I like to do for not just my PhD, but for my entire career, is to use this untapped knowledge of the non-coding RNA to both diagnose and treat a whole host of diseases that we didn't even know existed before. And lastly, I'd like to close that Emma is still receiving care today, and while a very effective cure is still underway, her family has helped other rare disease patients and families like hers find answers in community. And lastly, I'd like to acknowledge my lab, our collaborators, and also importantly, the patients and families that do this work. And I'd also like to thank the people in the RCTP, especially Kiki, Callie, and Trey for helping me become a better and more confident speaker. Thank you.